Hey there, welcome back to COG 266. Today we're going to finish our discussion of sort of introducing a variety of our sensory receptors. Last time we started with a discussion of how the heck does stuff get from outside in the world into our neural system, right? So if the brain is a system for accepting information from the outside world, perception, transferring and transforming that information, cognition, and doing things in response, action. The topic at hand is how does information from the outside world get into the neural machine works there? So focusing on perception in general, this is what we make of the outside world. The way that we construct our understanding of the outside world is the perceptual processing that happens in the early steps of the brain from the input stuff. Essentially, if you're a computer nerd, it's part of our API, right? It's one of the ways that we can request from world information. Now, remember, we need to take certain kinds of information to figure out what to do in order to both find good things and avoid bad things. It sounds like a super reductivist approach, but that's exactly right. I mean, you're an organism. You are a mammal, and you are able to interact with the world. Mostly you kind of want to survive. You might want to eat good foods and hang in good spaces. And you might want to avoid things like walking off of cliffs and getting eaten by tigers, right? So the only way a system that, up until this point, before we started talking about sensory perception, was just neurons talking to each other, somehow we got to get that in here. So what kinds of information? We've already touched on this, but just, refreshing. Worldly information out there. There's a lot, but the things that we're used to talking about and thinking about, physical contact, so how much pressure is being applied to me, how much heat is there, what temperature is there, what are the textures of things, so if I touch one kind of object versus touching a different object, what's the difference in pattern I experience, pain, how painful is an experience to me. We have sound, which has all kinds of stuff, pitch, timbre, the complex interactions of overtones, and I don't know music theory that well, but the idea of how sound waves all combine to make things like my voice distinct from somebody else's voice, right? Even though we're saying the same things, we can even be talking in roughly the same average pitch, but they are different, right? Different sounds. Light, we have all kinds of things from light. Hue, luminance, reflectance. And then there's chemicals, huge array of particles that we should probably beware of, and both the good and positive and negative beware, like beware, there is food nearby. Beware, oh, there's a skunk. We should probably avoid the skunk. Not a fun time. Now, just before we move on to recap some of this, remember that there are tons more kinds of information out there. In fact, if you think about the nature of the world itself and the many, chemistry, biology, physics, math, all this stuff, anything can sort of be information, right? Anything can be a signal compared to noise. So there's tons of other basic kinds of information that would be maybe interesting to be able to understand. Things like radioactivity, electrical currents, magnetism, all this kind of stuff. So just remembering that we are a system that's able to sense some things and other things go right over our heads. So remember, you are limited. So, okay, let's recap mechanical energy real quick. So, the way we capture mechanical energy from the outside world is through mechanoreceptors. These are the neural cells that have a particular construction that's capable of reacting to physical distortions, things that are changing in the arrangement of particles nearby, right? So, somatosensory system, that's touch, detecting pressure with most of our body, and the auditory system is sound, where we detect air vibrations in our ears. Now, I'm going to throw up a couple of recap slides. So there's a lot. Brace yourself. Okay, somatosensory mechanoreceptors, right? The one that we focused on mostly was the Pacinian corpuscle. and that is it has these bulbs, these myelinated bulbs, that when pressed upon, the cells will deform a bit. The membrane will deform, as you see here at the bottom. When the pressure is applied, channels open, ions flow in. Hey, we know what happens from there. Action potentials happen. And with Pacinian corpuscles, the amount of pressure you apply changes the firing rate. So the more pressure applied, the more firing rate there will be. So that recaps the basic of how the heck any one given neural structure could respond to something like touch. Touch happens, the change in information is exchanged, and now we're in neural land, right? Remember, neurons are the things we want to get the information into so that they can go and bounce around the rest of the brain. There are two kinds of somatosensory mechanoreceptors. There's the fast-adapting ones, which help us detect changes, and the slow-adapting ones, which help us keep track of sustained activity. You could understand how those could both be very important, right? In one way, I want to be able to feel that I am sitting down. But in another way, I want to know if my chair starts to break. So a sudden change, even though very small, so that the amount of continued pressure in the chair, if the leg starts to crack and move, it's not much different, but the change is important, right? I need to understand the change. Also, a handful of kinds of other kinds of mechanoreceptors that are in places like skin. We have free nerve endings, which help us detect pain. Ruffini endings, which help us detect things like stretching. Meissner and Pacinian corpuscles, which are texture and slow vibrations in the Meissner, and Pacinian does the more deep pressure and fast vibrations. And then Merkel discs help us with the sustained overall pressure type stuff. That's not, again, this is not a rote memorization exercise, but be aware that those are all related to mechanoreceptors. These are all kinds of mechanoreceptors in the skin that serve different functions. So that is touch receptors at a broad glance, the stuff that we've already covered. Let's also take a peek at auditory mechanoreceptors. Now, if you recall, I have a transition there for some reason. Your cochlea. Your cochlea is the spirally bit past your eardrum in your ear, and it is essentially the basilar membrane is the long, tough, rolled up bit that spirals into that, and all over the basilar membrane are hair cells, little neural cells with hairs attached to them, that because the basilar membrane is different thicknesses all along itself, it's going to vibrate. Different parts of it are going to resonate with different pitches of sound. That's just a fact of how vibrations happen. When air vibrates because of sounds, because of different sources of vibration, things around you will resonate at different frequencies. That's why you've seen the singing, the opera singer can shatter a wine glass, right? That's explicitly because the opera singer can reach the resonant frequency of that wine glass and make it start to wobble. Once it starts to wobble, it doesn't stay together very long. That is the same principle, but it's stretched out over this tough rubbery thing that's rolled up inside your ear. So different sounds make different parts of this rubbery boy vibrate. So following it pretty quick, the quick version of it is air vibrations will hit your eardrum. Specific parts of the basilar membrane will vibrate based on the pitch, and it can be more than one at once. Everybody has multiple pitches in their voice, for example. Specific hair cells along that membrane will vibrate in this potassium ion-rich fluid, meaning it's just going to absorb more of those potassium ions. It's just going to whip them back and forth across them, and that means that more will enter the cell. Ta-da! The Potassium ions enter the cell, creating action potentials, and then they go off to the central brain for processing to say, hey, what was that? Right? So we get the pattern on the surface from the auditory mechanoreceptors, and then the brain will take it and say, okay, let me try to make sense of what these vibrations might mean, and give us the sensory experience of sound. We are now understanding sound from this pattern of vibrations that we've been able to receive and convert into neural activation. Yeah, and also just the last note is that the piano metaphor helps a lot. So, low pitch will vibrate this area, whereas high pitch will vibrate to the right. The thinner it is, the higher the pitch the vibration goes for. Okay, so that's just to recap, kind of get us back in the saddle for sensing mechanical energy. So these are what are known as directly gated receptors. So they respond to the deformation of the cell membrane or the movement of the cell itself. Right? So mechanoreceptors being touch set receptors in the somatosensory system and hearing in the auditory system. So now we're going to talk about the other broad category of receptors, and that's the second messenger receptors, which include taste, smell, and vision, which is sounds a little odd. We'll talk about that. Let's start with chemical stuff, basic chemical stuff. So, chemoreceptors are, as you might guess, receptors that are able to interact with specific types of molecules to be able to detect exactly what you're dealing with in the chemical world around you. Right? So your gustatory system is taste. That's your taste system. And that's detecting chemical compounds that are inside the mouth, or if you lick something, that also works. Not always the worst. Helps to get a sense of, is that toxic or not? And then the olfactory system, which is smell, detecting chemical compounds in the air. So a chemical sense is able to respond to a lot of extremely specific chemicals in very complex and meaningfully different ways. Wild stuff sometimes. The subtlety of the chemicals, for example, of the things that you're getting from the air when your coffee has gone from just right, it has a particular set of chemicals released that give it a smell, to a bit burnt. You can tell the difference between those two. I don't know, not all of you are coffee drinkers, of course, but you may translate this to toast. You can kind of tell when it's a little bit burnt. And the reason you can tell that is because you have receptors that are very good at picking out carbonized stuff, burnt things. So let's talk gustatory receptors. Your taste receptors, so gustatory chemoreceptors, these are taste receptors. They're organized into taste buds. You've heard this, of course. And they have a wider range of configurations. So chemicals in the mouth are turned into a bit of a solution by the saliva. And then that solution, the chemicals in that solution, can then bind to your taste receptors. So just right off the bat, everything is chemicals. Your chemicals, your neurons are chemicals, your body is chemicals, your food is chemicals, everything is chemicals. So the idea is that you're getting the particles. You're able to take particles onto the tongue and detect things about them. Now, interestingly enough, taste receptors are kind of all over your system. They're all throughout the digestive system. You don't always have the explicit experience of, I am tasting something in my stomach, right? But you can kind of feel like, ooh, this thing doesn't agree with me, right? And even in the lungs have a little bit of taste reception. Not sure why. But something about taste receptors is that they die and respawn generally. I say respawn, it's a gamer term, but they regenerate pretty regularly. So these are not fixed. In fact, they say, this is one of those like sort of social maxims, but it's the idea that every 7 years you have a different set of taste buds. I don't think there's a lot of science about that, but the idea stands that you're not always going to have the same set of receptors. So things could change. Things can gradually change in your experience. So let's look at some of this. Here's a tongue. It has a variety of little bits and bobs that are essentially all different kinds of taste buds. If you zoom in a bit, we can see in a taste bud, we have little taste hairs, and we have then additional cells that are prepared to receive. So things will bind to the hairs up there, the little taste hairs. They'll bind up there, the chemicals will bind there, and they will send stuff forward that is then caught by the other cells later, the basal cells. Let's talk about the sensations of tastes, though, before we dig further into that. There are 5 basic taste sensations: sweet, bitter, sour, salty, and umami, which you could also list as savory, but it's good. Sweet, bitter, and umami are all going to activate receptors that result in G proteins that send messages to secondary receptors, which will pass the information forward. So the idea behind that is that kind of like we saw with neurotransmitters, right? They will bind, and they don't actually change the ion flow. They send some kind of protein off into the next cell that will do some work to change how that cell works. Instead, in this case, it's not about changing anything. It's just about detection. So certain molecules that are associated with the sweet, bitter, and umami flavors have these secondary receptors going on, whereas others are a little bit more direct. So sour and salty activate ion channels very directly. Why might this be? It's not perfectly understood, but one going idea is that these acids that are associated with sour flavors and salts have plenty of ions to go around, so they just can, right? It would be quite useful to just be able to pick those things up quickly. The more advanced flavors have more to do with more complex molecules, need something else to sort of mitigate this scenario. Let's look at that. So here's a salty taste bud. Here's a salty receptor. Salt comes right in, and it changes the... So step 1, there you see the eye, right? Salt will enter right in and change the ion flow to allow more ions to come in, and things will then cascade as we already understand them. Again, you're not here to learn all these precise mechanics. The idea is just that in step 1, the salt will just enter the cell, and then that will cause activation to occur. Whereas in a sweet receptor, for example, the molecules of the sweet don't enter the cell. They activate on the outside, and again they cause our friends the complex and mysterious G proteins to be activated and go cause some kind of change in the cell that will then trigger the action potential. So the difference between these two is that in the salty cell, salt will enter and just directly change the charge of the cell, whereas in the sweet cell, the complex sweet molecules that are on the outside only change something within the cell. They don't actually enter and mix in with it. So I hope this helps to illustrate how we can take chemicals in the mouth and convert them into signals, right? Again, it's all about how we can generate an action potential from some kind of outside force. Other things about taste cells, there is no neural specificity. They can respond to one taste or lots of different tastes, which is a little weird. You typically think of neurons as responding sort of in a specified way, right? Or as these receptors in responding in these specified ways. But the key is that any one cell, any one taste cell, it's not going to actually produce a meaningful taste sensation, because as with many of these things, it's about the overall pattern of taste and the experience of taste in the cells across many cells, right? So you have to have a lot of flavors going on all over the tongue for the complex experience of taste to take shape in your brain. You're not going to get a flavor out of one taste cell activating. So that's the idea. Chemicals come in, they flow in. Some of them go directly into the cells, and some of them go more the more complex chemicals will just activate the cells. Either way, that's how we taste things. Now let's talk olfactory. So, olfactory chemoreceptors are the ones about smell. Olfactory is smell-based. These are located in the upper membranes of the nasal passages, so way up, very, very close up to near the brain. And they kind of short-circuit this common perception we have that a lot of stuff reaches the brain by way of the brainstem and such. These go right to the olfactory bulb on the base of the brain. So these are, in many ways, our most ancient structures. And it makes a kind of sense. If you think about an organism that must interact with its environment, being able to take things in chemically from sort of the surrounding environs, that seems pretty important. So these are very tightly tied to brain activity, because they connect directly up there. I mean, everything is brain activity, of course, but what I mean to say is that they have a pretty direct line into their respective olfactory bulb in the brain. So, what are these receptors like? Well, they have some things in common with taste receptors, but their method is a little bit more like the molecules in the air will get captured in the mucous membrane inside of your nose, your nasal cavity, and cells will extend like hair into that mucous membrane, and similar to sweet and umami and what was it, bitter, any chemical can bind to these hairs, these cilia, and cause G protein production, just like in taste, right? So the hairs are free floating. Particles are going to get stuck in the mucus, and they will touch these various olfactory receptors, and they will send the message on that, hey, this thing's here. I found this chemical, right? And again, here's a bunch of stuff. Odorant hits, the receptor activates, the G protein moves, and then the channels move, and then, hey, activation. I hope these diagrams are seeming less and less dense every time they pop up. You know, I don't know the exact role that adenylate cyclase plays, but it is one of the pieces that causes the action potential between the odorant activating the cell, the G protein moving forward, changing on the right side there, changing the ion channels to make them activate, and ta-da, this neuron, this olfactory receptor is sending the message that this thing is here. So, here's the trick. Whereas taste receptors are not specified almost at all, they're more just kind of summing up base flavors that we use to combine into more complex flavor ideas, olfactory receptors have a huge variety. There's like 400 different kinds of olfactory particles, particulate in the air, that humans can detect, specific. And again, this is kind of makes sense because you think about the ancientness of these structures, how far back they go. And it kind of makes sense that we would need to develop very specific detectors for all kinds of stuff as it became more relevant in our environment. Now, we're not especially cool here. Other species have tons more than us, way more, millions in dogs. That's why dogs are so obsessed with sniffing the ground. In fact, I want you to think about that for a minute. If you could detect millions of different kinds of particles, you'd probably have your nose in the ground more often to figure out what the heck's been going on around here. So whenever you see a dog get all sniffy, remember that their mind is lighting up with all kinds of information that you will never have access to. So have a little sniff empathy for dogs. So each one of them is going to respond to different odorants. And there is some overlap. They can respond to similar odorants too. That's how we can get a lot of sort of gray areas, and how we can capture things that we haven't developed very, very specific odors for. So whereas a dog might have millions of distinct receptors, we can still capture some of that detail. It's just more about, oh, well, these 10 different receptors are lighting up. There's some kind of chemical here we're not sure about, right? And so combining these 400 different receptors, we can discriminate about 10,000 different distinct odors. That's a lot. That's a load. But think about it. How many things can you detect in different ways? Strawberries versus raspberries, very slightly different, but there they are. You can kind of tell. Blueberries versus toast. Skunk versus burnt rubber. Well, that actually kind of can be confusing sometimes. But that's the idea. Another interesting note about the olfactory bulb and the olfactory system is that it's tied very tightly to memory. So the olfactory bulb is connected pretty closely to areas of the brain associated with memory. This is why you may have noticed in your life that smells can often elicit very strong memories. You might, the tragic one, the smell of an old partner. They used to use the same perfume. But the idea is that there's a reason that that this is so powerful. They have this direct connection. They are physically and connectorally close, right? These systems are near each other. So the standing question is, why might that be useful? Well, one thought is that memory is a pretty useful thing for an organism, for any organism. In fact, it's one of our coolest tricks. It's one of our most important tricks as a cognitive system, to be able to hold on to something for later. And if this olfactory system is one of our most ancient tools for detecting the outside world, then it makes a lot of sense that it would be tied tightly to memory, especially because in early life, chemicals are pretty much the key thing, right? I need to know where good food is and where bad acids aren't, right? So that I don't have to get dead from any of that. So, yeah, that's just the note on the olfactory bulb and memory. Now, one thing I want to bring up is this idea. What does natural gas smell like? It's kind of bad, right? If you've ever left a stove turned on or whatever, you get this kind of stink out of it. And you're like, ugh, the natural gas is on. Or maybe you've walked by a area where there's been like a leak in a line and you can smell it, right? Natural gas it can be troubling in that way. Here's the truth, though. We are limited, right? So if one of the goals of this system is to be able to avoid bad stuff and seek out good stuff, but in this case, avoid the bad stuff, some things we actually can't detect. Natural gas, to us, is completely odorless by nature. So if that gas line were open, you would not be able to tell, because just the way it comes out of the ground, the way that we actually harvest natural gas, we wouldn't be able to tell. So this sucks for us. This is bad because it's incredibly dangerous. A free-floating chemical in our environment that can smother us, poison us, and explode us. That's not great. We artificially add that rotten egg smell so that people will notice it and be able to avoid it. In fact, we give it a bad smell because we don't want people to think, oh, good, sweet candy. We want them to say, ugh, yuck, this sucks. I want away from this, right? So that's one way that our system is limited. We can't detect this thing. We don't have any receptors that bind to natural gas particles, but we do have the ability to, when we use natural gas, add bad smells in to help our systems deal with it. So that's the idea of the chemical senses. We're capable of detecting a lot of fairly complex molecules, which is very cool. Now, remembering that these are very old senses, they come from our chemotaxis friends, the oldest way we have of cells communicating with each other in the outside world. And it's important to note that they connect to really old neural and behavioral systems, so survival, sustenance, nutrition, and memory. An interesting feature of chemical senses, though, is that they're also really subjective. So odd things like tastes, your experience of taste can interact a lot with weird things like visual qualities of the food, textural qualities of the food, the temperature. A cold, there's a reason we don't microwave grapes and eat them. We don't really find that to be delicious to our senses for whatever reason. Even though it's the same chemicals, they've just been excited for some reason, for whatever, for that cause. So this is why it's a fascinating thing that this is the case. This is why when people talk on cooking shows about the presentation of the dinner plate matters so much, it kind of does, because all of these other signals are going to go into your experience of the meal along with the taste and the flavor. You can't just eat, you could just eat a protein slurry, but we choose not to. We choose to prepare things in different ways. You could throw all of your food in a blender, but you don't, even though it would be so easy to eat it, right? I don't need to put a hamburger in a blender. In fact, that sounds terrible. And that's kind of the point. Why does that sound terrible? Well, all these other elements come together to make the experience of flavor a little different. It's more subjective. It's more impressionable to the outside world. Just something to think about. Just something to keep in mind. So, now let's move on to a different kind of energy. Photoelectric energy. Light. This is an interesting use of second messaging. So, photoreceptors are cells that can be disrupted by photons hitting them, which then allow us to detect the details of visual environments around us. So this is the visual system. Vision is the sense, and detecting light with our eyes' retinas. Let's talk about how these weirdos work. So, visual photoreceptors. There's only a handful of kinds, and they are distributed across the retinal mosaic at the back of your eye. So down there at the bottom, you have the back of your eye is where the retina lives. So the light comes in. It's actually it's twisted upside down because it comes in through your pupil and goes to the opposite side. You have at the back of the eyeball the optic nerve. And then it's like the cable, right? It's like the Ethernet cable from your eye. And then all across the back circular area of your eye is your retina. And the idea is that there are all of these light receptors waiting for signals, a little bit like a camera sensor. You can see on the right there, that is sort of those are what they actually look like, the cells themselves, the photoreceptors. And this is sort of a pattern of showing, in the middle here, we'll zoom in on that, showing roughly how those sensors are laid out. And there's different colors to represent where different neural, there are multiple kinds of sensors, and they have different colors in this image. It's not a literal, it's not like an actual photo of the eye. This isn't how it looks, but this is more to give you the sense of how these sensors are distributed throughout the back of your eye. So, what are these sensors? Photoreceptors. So, photoreceptors, they produce a neural response when they are stimulated with light. The way that happens is a little bit wild. Light hits a protein, rhodopsin, on the receptor's surface, which will cause a structural change that sends G proteins on, right? We already know a little bit about G proteins and how they are sent forward to activate other stuff. So the fact that rhodopsin is unstable and light hitting it makes it kind of shake loose. It sort of changes, and that causes activity. It causes a G protein to run off and say, ooh, I'm going to go activate some stuff. One oddity is that it's reverse of what you might think. Darkness increases the receptor potential, while light will actually decrease it. If you think about that for a moment, it might seem wild that your neural receptors are most active in the dark, but ultimately, if you think about it a little bit more, it doesn't actually matter, does it? The only thing that matters is the difference, right? So if light has one, if receiving light has one signal and not receiving light has a different signal, that's all we really need, isn't it? So, what's their response like? Well, there are a couple features of the response of one of these photoreceptors. There's a little bit of a startup process, so we get the best of both worlds like we had in touch. We can detect changes in light, similar to the way that we can also detect, or similar to touch, we can also detect sustained light, right? So just like touch, we can keep track of the onset of a change, but also hang on to the signal and keep track of what is the actual activity happening here. So there are two broad types of photoreceptors: rods and cones. You may have heard of rods and cones in your eyes before. Rods, about 100 million of them, mostly in the periphery. You have tons of them. And you can see them here highlighted. They're a little bit bigger. They're the thick ones. Rods we'll talk about how each of them receive in a minute. But there's a lot of rods all around the periphery, and then there are less but more concentrated amounts of cone cells in the fovea, which is sort of the central area of whatever you're looking at, right? There is that tiny region right at the back of the eye, right in the center. The things that you are looking at to take in what's going on when you read letters off of this screen, that stuff is hitting your fovea. You're not doing that with your rods. So cone cells, there's three major types, and they respond to a little bit different frequency of light. So there are some that respond mostly to red light. They'll still respond to things nearby, like kind of orangish and all that. But there are some that respond primarily to red, some that respond primarily to green, and some that respond primarily to blue, primary colors. So combining those signals, a certain amount of red, a certain amount of green, and a certain amount of blue, gives you a good sense of the color of things in the environment. Now, one note I like to give this to everybody: a practical home experiment you can do. You have your whole retina that is able to, take in. This is all of the area of your eye on the back of your eye that is sensitive to light. You also have your fovea, which is the more concentrated area of cones that are going to give you higher amounts of detail in your vision. So the whole retina and it is mostly rods, and then the fovea has all the cones tightly packed together. A way you can sort of imagine this for yourself is that your fovea is about 2 degrees of visual angle, and it turns out that according to basic anatomy, on average, if you hold your thumbs out at arm's length, hold your arm out at its full length, and then put your thumb up, That's about 1 degree of visual angle. So if you want to know how big your fovea is, put your thumbs together and look between it. That's what you're dealing with here. That's the amount of sharp detail you get in any given moment from the world around you. It does not feel like that because a whole lot of your brain is filling in details and coming up with other stuff. But the fact is, most of your cones, the detailed part in the fovea, are within those 2 thumbs at arm's reach. Just useful. Think about it. Party trick. Once we can party again. So, that's retinal cells. And that's just me redetailing this. So you got 6 to 7 million cones, mostly concentrated in the fovea. That central spot in the eyes that covers that little bit, about 2 degrees of visual angle. Both types of cells are going to respond to light, but there's a fundamental difference between them. There are many rod cells can connect to a single neuron, while a single cone connects to a single neuron. Cones are more detailed, and they each one connects to a single neuron, whereas rods can be kind of spread out and accumulate more signal from a distant area. So let's go over some of the implications of that. In rod cells, weak signals can be combined together to fire, meaning that you need less light to activate a rod cell, or rather a neuron that is attached to many rod cells. If you think about that in terms of neural signals, that makes sense, right? The neuron is going to accumulate a lot of signals, and if I'm getting a very weak signal but from a lot of places, then that means we'll be able to activate without that much light. That means rod areas are more sensitive to low light conditions. But it also means that rods are going to be poor at discriminating spatially different signals, because they can't distinguish, if all of the rods are always summed together, there's no way they could possibly distinguish between, hey, did it happen on the bottom rod or the top rod? No way to tell, right? So rods are bad at detail, but they're great at low light and sudden changes, right? Whereas cone cells, the signals remain separate, meaning that they need more light to activate, but that also means they are good at distinguishing the small spatial differences, right? It takes a lot more light to light them up, but they are, you can tell exactly where each one came from. This is why, it takes a lot more detail to extract good detail from the environment. You need to put a lot of neural circuitry into that. And that's why we only have the one little foveal area. It is useful to have light detectors all over the place, so we've got the rods all over, but to get the detail, we had to spend extra, so spending extra neural circuitry on it. So, using these rods and cones, just example things, which one do we use to navigate dark rooms? Rods are going to do more of that work, right? Rods sample that broader range, so they pick up enough light sooner than a cone to make sense of the room. You don't necessarily know what color the table that you just almost bumped into is, and you don't know all of its details, and you can't tell exactly everything about it, but you can see it in your periphery easily. Whereas, why are the cone cells concentrated in the fovea and not in the periphery? Well, because when we're focused on information, we're going to hold our gaze constant. So details like color and stuff, they're most useful where we're actively looking. Whereas that peripheral stuff is better spent on the sort of fast-acting, rapid-changing, non-focusy stuff, right? So rods and cones, 2 very different photoreceptors, but they are doing similar jobs to help us detect light in the world. So that's second messenger receptors. They don't involve the direct manipulation of gates so much, although I guess taste has a little bit of that going on, but it's not about it's not direct gated, right? Something still comes in. Instead, the external stimulus will affect the cell chemically, triggering activity that will then cause the firing to occur. So in the olfactory and gustatory senses, those receptors will interact directly with the chemicals, whereas in light and vision, light causes the release of internal chemicals, which then carry on in the system as above. So let's return to this thought on how does light get inside of our cognitive system. Remember our traffic cone, or not traffic cone, or traffic light, the information that comes in and the transformed version of it at the end. We have red, position highest, wavelength so-so, danger stop, right? We have middle, the yellow light, caution slow down, and we have low, the green light, safe go, right? What we do with this information is the light will strike 3 different types of color receptors on the retina in the cones, right? This will react with the proteins and send G proteins to second receptor neurons that will then sum up the information received. And at that point, we're in. Now, this doesn't get us all the way to transformed danger stop from red, but the more complex processing can now follow. For example, the most basic one is we have to combine the activity from the 3 different types of cones to get a sense of what colors are, right? I couldn't tell that green there was green unless I had a low signal from the blue and the reds and a high signal from the greens, right? I mean, that's a form of combination and a form of transformation. We turn 3 distinct patterns that we get from the cones into one experiential pattern of, hey, that's green. Interesting. So that's sensory receptors. We have the direct ligated receptors that are going to respond to deformations of the cell membrane. So our mechanoreceptors in the somatosensory system with touch and the auditory system with hearing. We have the second messenger receptors that are going to respond to chemicals, both internal and external. So that's chemoreceptors in our gustatory system, taste, and our olfactory system, smell. And then we have our photoreceptors in the visual system for vision. So there it is. These are our primary senses, the ones everybody talks about. It's pretty cool. Overall, we've got these mechanical, chemical, and photoelectric senses. We are able to detect that kind of information out there in the world. That's great. That allows us to do all of the things that we do. All of the cool, fancy, amazing, horrible things that we all do are only possible because we're able to sense the world. They all take this stimulus from the outside world and turn it into a neural signal, and that allows us to perceive the world and take actions on it. And then we do things, send it off to complex, fun, interesting places in the brain. And that's it. That's what you need to know about senses in a general sense. Now, we will dive a little deeper into some of the cool bits about how vision works at minimum. But from there, that's more of an exploration of how specific kinds of neural circuits all kind of come together. So that's it for now. As always, here's a final thought. Some animals have senses that reach beyond our own, such as being able to detect shifts in activity in the Earth's magnetic poles or electrical activity in the nearby water. Think about other kinds of energy and information available in the world. What might be a good kind of external information for our system to add to our repertoire of basic senses? All right, that's it. Thanks, take care.